talk of our Leaders Initiative Speaker Series as part of our MADRC. And the goal of this series is for early career faculty who are involved with our Leaders Initiative to share their research, to host other speakers who conduct research related to dementia or caregiving, and to promote research collaborations. And we're very excited to have Drew Bender as our first speaker today.
I'm going to introduce Drew real quick. Drew Bender earned his bachelor's in psychology from the University of Arizona, followed by several years of working in industry as a programmer and project manager. He then returned to the academy to earn a master's in psychology from San Diego State University, which sparked his interest in neurocognitive aging research. Drew's formative training was as a Ph.D. student in cognitive neuroscience of aging under Naftali Raz at Wayne State University. Following his PhD, Drew accepted a postdoctoral position with the Center for Lifespan Psychology at the Max Planck Institute for Human Development in Berlin. Studying a broader population-based representation of aging, he worked with the Berlin Aging Study 2 and research groups in quantitative methods and lifespan memory. In 2018, Drew joined the College of Human Medicine at Michigan State University as an assistant professor in epidemiology and biostatistics. Since 2021, he has been a co-investigator on the neuroimaging core of the Michigan Alzheimer's Disease Research Center, as well as a member of the executive committees of the MSU Alzheimer's Alliance and the Michigan ADRC through their Leaders Initiative. Without further ado, I'd like to turn things over to Dr. Drew Bender.
Unmute first is always a good step. Thank you, everybody. It's really a pleasure to be here. And thank you to Courtney and Stephanie for all their help in making all this possible and coordinating this. So I'm going to share my screen, and I hope you're able to see that. So today I'll be sharing some work from across my career on white matter changes in healthy aging and dementia, and just a brief outline of the talk today. I'll start with just giving a brief overview of my role in the MADRC, and then share some of the background in white matter and cognitive reserve literature, and why I think there's a case for white matter as a potential substrate of cognitive reserve and resilience. Some of my early findings that have led to this in aging and memory and healthy aging samples, as well as some emergent challenges and opportunities from Thad Polk and work with him in the Michigan Neural Distinctiveness Study, and more recent findings from the Berlin Aging Study 2 and the University of Michigan Memory and Aging Project that most of you are familiar with, or UMMAP, and finally some emerging questions and some future directions that are currently under work. And I'm happy to take questions during the talk. Feel free to unmute yourself, or if you put them in the chat, I believe Stephanie has graciously offered to relay those to me. So in the MADRC Steering Committee Leaders Initiative, so my role has been as a co-investigator on the neuroimaging core since its inception a couple of years ago. And this has included consulting on diffusion MRI sequences and analysis, and consulting with internal and external investigators and students on image processing, especially relating to structural and diffusion magnetic resonance imaging data, and also sharing process data with center investigators, which is currently underway. Scott Peltier and I are currently working to facilitate the transfer of all the work that I've been doing so that these data and resources are available to center investigators. And this also has involved the development, validation of a number of computational processing pipelines so that we can take raw neuroimaging data from the scanner and put it through a host of different processes in order to get some final endpoint of data that is usable in aging and dementia research. And this includes advanced diffusion MRI modeling ranging from diffusion tensor, free water elimination, methods like neuro-orientation diffusion dispersion imaging, or NODI, and constrained spherical deconvolution, or CSD. Also the creation of brain imaging templates, which are really useful for more population imaging so that we can do mass univariate analysis within brain imaging data, and more recently work with my graduate students in structural connectomic analysis of white matter network properties that are known to change in aging and in neurodegenerative diseases. And so a lot of this work I also want to acknowledge, besides the role that I've had, has been really supported by the MADRC, and this began before I was a co-investigator on their neuroimaging core with a pilot research award, which is now a development award, and that was matched with funds from the Pepper Center, and is looking at white matter complexity and practice effects as novel markers of MCI and dementia. I'll talk about this a little bit later in the talk. I've also been a co-investigator on a development project award with Norman Scheel, who I believe is on the call, a postdoc in MSU's Department of Radiology, for investigating dimensional complexity and functional MRI BOLD as a marker of early cognitive impairment. And finally, I've also been the recipient of a REC Early Career Training and Dementia Research Award. This has been also truly transformative, working with Ben Hampstead as the primary mentor, as well as with Irving Vega, Laura Zahodny, and Naveed Siraji to really facilitate my growth as a dementia research. We're specifically utilizing this, and we're about to get started. We have our data use agreement now approved for piloting smartphone-based cognitive assessment measures in UMMAP participants that we'll be doing remotely from our lab at MSU. So that's where I'm coming from, but the conceptual and research perspective, I think, benefits from some further background. And so just to start off is the cognitive reserve framework that's been popularized by Jakob Stern at Columbia suggests that if we look at the relationship between cognitive status here on the y-axis and Alzheimer's neuropathology on the x-axis, that we can set some threshold for dementia below which cognitive status falls underneath some critical and clinically diagnosable level. And so somebody who has some level of neuropathology over time may withstand that and eventually start to decline until they succumb below that threshold for dementia. Other people, in contrast, may have much more prolonged preservation of cognition despite ongoing neuropathological processes. And so we can think of these two individual cases as representing low reserve and high reserve. And so high reserve is believed to reflect ongoing maintenance and compensatory processes that buffer the cognitive system against neuropathological influences. And so we can think about or define cognitive reserve then as the preservation of normal cognitive function despite brain damage or despite apparent neurodegeneration or neuropathological processes. And traditionally, these neurodegenerative processes have been characterized as a function of cortical and subcortical brain atrophy, degeneration of white matter, as well as the presence of amyloid and phosphorylated tau pathologies. And it's really critical to note that the presence of cognitive reserve, or those with high cognitive reserve, also tend to have higher earlier life educational attainment and to partake in ongoing cognitive engagement through occupational, recreational, and social activities. It's also worth noting that cognitive reserve is correlated with the maintenance of cognitive plasticity, the ability to learn through incidental exposures. And often this is reflected in practice effects across longitudinal assessments of cognitive performance. However, there are no established neural mechanisms nor markers of cognitive reserve to date. And this has become a really strong interest for me, and especially the role of white matter degeneration, because traditionally white matter degeneration has been viewed strictly from the perspective of white matter lesions, here shown as bright or hyperintense regions on a T2 weighted MRI flare image and around the horns of the lateral ventricles, as well as in some deeper white matter regions. And these are associated with vascular disease, metabolic dysregulation, and often are associated with executive and sometimes nestic dysfunction. But in reality, we need to think about what's happening not just within those lesioned areas, but across the whole brain's white matter. And so this cartoon schematic from over Pasternak at Harvard really illustrates some of the larger system at play. And so we can see areas where pathogens or autoimmune dysfunction or trauma exert some, convey an inflammatory response from microglia, which release cytokines that have a degenerative process that is pronounced with regard to myelinating oligodendrocytes, which form the myelin sheath around axons. And this degenerative process reduces the packing density and coherence of white matter axons, which results in a reduction in anisotropy, or the coherence. It also then triggers further inflammatory responses from microglia, and together this also results in some atrophy of white matter axons and an increase in the extracellular space, which Pasternak refers to as a free water increase. And so this is the general mechanism that we're observing, but it's also worth noting that not all white matter is equivalent. And so the latest developing white matter fiber systems, the association fibers that connect regions within a hemisphere, that connect cortical regions within a hemisphere, are the latest to develop, tend to have smaller diameter axons, and tend to be more vulnerable to Alzheimer's pathology, as well as age-related vascular pathology and metabolic dysregulation. And this is in comparison to somewhat earlier developing commissural fibers that connect the left and right hemispheres to one another, which are also affected by Alzheimer's pathology, but sometimes a bit later in the course, versus projection fibers, which connect subcortical regions and cortical regions within a hemisphere. Projection fibers are the earliest developing, tend to have large, more uniformly organized fibers of larger diameter. And so this notion that the earliest, the latest to develop are the earliest to decline is captured in the last in, first out hypothesis of brain aging. And I also want to call your attention to the fact that we can represent these same white matter fiber tracts really well using magnetic resonance imaging, or MRI, using diffusion weighted MRI and tractography, which really represents these same fiber systems and permits all sorts of additional analyses. However, if we look at within this square, we can see that there are fibers from projection regions, as well as, the projection fibers kind of in here that cross over these association fiber regions. And so we have some regions where there are combined differential effects of earlier and later developing white matter fibers. So diffusion MRI is based on the movement of water molecules in tissue, and that it moves depending on its barriers. And the myelinated axons, so myelin is hydrophobic and it serves as a barrier to molecular water diffusion. So water should move preferentially along the length of the fiber bundles, and we can characterize that tendency towards uniform or coherent diffusion as a function of anisotropic diffusion, as a measure of non-randomness or coherence of the fiber bundle, in contrast to isotropic diffusion that we'd observe, for example, in the lateral ventricles. And if we sample how water is moving in a whole lot of different directions in space, we can summarize this using some kind of mathematical function like a tensor, so represented as this ellipsoid in space. And so when that tensor tends to be more spheroid, it's more isotropic, and when it's more prolate or cigar-shaped, it's highly anisotropic, and we can infer different information about white matter, microstructure, and organization based on the presence or absence of an anisotropic signal. And we demonstrated longitudinal support for this last in, first out hypothesis in fractional anisotropy, a measure of anisotropy in white matter, showing that over time in middle-aged and older adults, over 7 years, that association fibers seem to show a more monotonic and pronounced decline relative to commissural fibers, which tend to show more of a decline after about age 70, and relative to projection fibers, which tend to be more robust or resilient to the effects of age over time. However, it's also really critical to note that not all effects across the lifespan on white matter are negative, and that white matter also undergoes remodeling and has considerable capacity for plasticity. And so when we give someone a specific training activity, like teaching somebody to juggle, right, or in preclinical rodent models, if we use some kind of reward training condition, that we can actually see improvement over time, that these behaviors, this behavioral plasticity, is associated with myelin remodeling and the recruitment of oligodendrocyte progenitor or precursor cells, as well as the expansion of existing oligodendrocytes to expand their myelination on axons that are associated with cortical regions with higher activity as a function of training. And so turning back to this cognitive reserve perspective, when we think about something like that, the fact that cognitive reserve is correlated with educational attainment and with ongoing activity, cognitively engaging activity and social activity that requires updating of information, it really begs, in my perspective, some different thinking about white matter changes as opposed to simply thinking of them as monotonic indicators of decline. And so the new research perspectives that I'm operating under are the notions that white matter microstructure and network connectivity may be critical mechanisms underlying cognitive reserve, or the ability to withstand the negative effects of pathology, and that lifespan changes in white matter microstructure then reflect both adaptive and pathological influences. And we can think of white matter organization then as changing adaptively across the lifespan in the absence of clear pathology to support ongoing maintenance of cognitive abilities. And the idea that formal learning and lifelong engagement further modify white matter microstructure and networks, that it's not simply early life educational attainment or exposures, right? But that something that may set the stage for. It's also critical to, I think, acknowledge that individual differences in genetic, environmental, and other life course factors moderate these effects, the beneficial or adaptive effects of white matter in supporting cognition. So how did I come to this perspective? Because it is somewhat unconventional. So some of my early work from my dissertation, where we looked at about 100 very healthy adults from 19 to 79 years of age, and look at change over 2 years in white matter. And here I'm going to show you anisotropy measures. And we used a region of interest approach. So we're actually capturing a mean value from different probabilistically identified tracts and sampling the FA values from then. And we used a latent change score modeling. I won't go into all of this, but if there are questions, I'm happy to talk about it. But one of the key features is we can actually quantify and do a significance test on variance in change to know whether people are changing at different rates from one another. And when we do this, we actually see mean increases shown here in blue in regions like the body of the corpus callosum and cortical spinal tract or posterior limb of the internal capsule, and mean FA decreases in other regions shown in red, like the genu and forceps minor and the corpus callosum and inferior association fiber tracts. We also saw significant variance in change, so people changing at different rates from each other significantly in most of these same regions. And to exemplify some of the heterogeneity that we observed in change, I'm going to show you from the projection fiber regions. So the anterior limb of the internal capsule and posterior limb of the internal capsule that we can see here on this dissection. And we see in the anterior limb, there is considerably a lower magnitude of change and more variability in change, whereas the posterior limb, we actually see mean increases in FA over time. And one of the reasons that we think this may be happening is that the anterior limb has white matter fibers. This is an electron microscopy image from Axer and Kaiserlink's work from 2000. These fibers are oriented in multiple different directions, so they're connecting different regions, whereas the posterior limb has axons that are of a larger diameter and more uniformly oriented. And so the question becomes, how much of what we think about change in FA is really reflecting some differential changes in the underlying crossing fiber populations relative to the largest axons that seem to dominate the signal that we're observing. And we followed this up by looking at how these changes in white matter related to changes in associative memory over 2 years. And we were looking at a composite measure, a latent composite measure of associative memory, including continuous learning and recognition. And we saw, as observed in this scatter plot, here we're comparing baseline scores for FA on the x-axis and baseline scores for memory on the y. And as expected, we see that higher baseline FA is associated with higher baseline memory. Cool. However, when we looked at change relationships, we see here 2-year change on the x-axis in FA and 2-year change in memory on the y-axis. We see that greater longitudinal change or greater longitudinal increases in FA were associated with lower change, or actually more declines in memory, and vice versa. So those who actually had more negative change in FA actually had greater retest improvements in memory over 2 years. And so to further probe this, we created what I call a spaghetti and meatball plot. And so the dots that you see here are the same as the plot on the left, reflecting the cross-sectional patterns of relationships. But the lines here, the length and direction of the lines reflect the magnitude and direction of the 2-year change score, right? So we can see that the cross-sectional pattern essentially becomes the opposite of the pattern of change-change associations. But it begs the question, FA, which has been described, so fractional anisotropy or FA, as a measure of white matter integrity, it's totally counterintuitive. So why would 2-year FA declines be associated with memory improvements? And this is sampled across a number of different white matter regions. Well, it warrants thinking about what FA is really capturing here. And so crossing fibers can affect FA. And so in these regions where we see mean change FA increases or decreases, so if there are regions where we have uniform numbers of fibers with the same signal in both directions, and one of those gets added, then FA is going to increase, right? So there's a preferential increase of one of those crossing fiber populations. However, if the opposite pattern is there, right? So if one of those is stronger, has more fibers than the other, and the one with the fewer, with the lesser signal, with only, for example, here the vertical orientation of fibers gets an additional fiber added to it, or a stronger signal, then FA goes down. Okay, well, why across the lifespan would you have an increase in the signal from these smaller orientation of fibers? Well, some elegant work by Yang and colleagues published in Neuroscience in 2013, they looked at the effects of environmental enrichment on myelinated axons in middle-aged and older rats. And their environmental enrichment condition involved the reconfiguration of the physical environment twice a week, which essentially necessitated a constant updating and learning condition. So it's not just the presence of enriching environment and stuff that the rats can utilize, but actually forcing them to constantly relearn their environment. And this was associated with an increase in mean axon length and mean axon volume, but no effects on the total myelin diameter, which was interesting. However, they actually did find that environmental enrichment induced increases in myelination preferentially in the smallest diameter fibers, shown here in yellow. And so this may be the mechanism why we see these differential changes in FA. And the reality is, when we look at diffusion MRI, it's not microscopy. Most white matter fibers contain, or voxels contain crossing fibers. So in work from Ben Urison in Antwerp, he analyzed the number of crossing fibers in brains, and it's exemplified here. So regions with red have a single orientation of white matter fibers, green have 2 orientations, and blue have 3 orientations. So a single orientation of fibers shown in red is really the exception, not the rule. And so when we look at a tensor model, if there is a single orientation, the tensor model does a nice job in capturing the nature of organization. But when there's more than one orientation, suddenly we get this planar response, which is uninterpretable. And so put differently, so diffusion tensor imaging, when we have those planar effects, if we try to use it for something like tractography to see where is one region connected to, we get a relatively sparse representation of those fiber tracts in the brain. So this problem is what's really prompted the development of alternative methods, including constrained spherical deconvolution, who wanted to know, how can we model white matter crossing in crossing fiber regions? And so constrained spherical deconvolution actually fits a response function separately to the underlying orientations, so that we can actually get a measurement that captures the underlying orientations of white matter fibers in the MRI voxel. And when we use that for something like diffusion MRI tractography, in comparison to the tensor model, it gives a far more robust representation of these white matter tracts. So shortly after I joined Michigan State, Thad Polk, who I had known from as a visiting scholar at the Max Planck, we used to run into each other in the bakery around the corner from the Institute. He reached out to me because he had a project led by an undergrad and a postdoc in diffusion MRI data from the Michigan Neural Distinctness Study. And as a part of that, I said to John Plass, the postdoc working on this project, it would be really cool if we could somehow model the complexity of these underlying fiber populations instead of just trying to rely on FA or other tensor measures. And said, well, we're going to use this new advanced method from MR-Trix. And actually, and he looked into it, he said, actually, they have a method that can output complexity of crossing fibers. So I said, cool, let's check this out and see what's going on there. So, and this is a community sample of 25 younger and 45 older adults. And one of the cool things that we can do with this method is, this is called a fiber orientation distribution, or FOD. And unlike a tensor, it has a lot more information which we can get from it. So we have individual orientations. Here I call them F1, F2, and F3, and this is arbitrary. So F1 the largest, F2 the second largest, F3 the third largest. And the length of each of these peaks or lobes of the FOD, we can take the integral of that and get a measurement of fiber density. And so here at the top, we have a representation of a normal fiber bundle. And if there is atrophy or decline in myelination, there's changes in fiber packing density, which we could see as represented down here on the left. Or if there's a total loss in the caliber of the fiber bundle, we'd see a loss in fiber cross-section. I'm going to mostly focus today on fiber density. And we can also take the orientational information from the FOD, so that we can actually look at what is the specific orientation of fibers and encode the information for fiber density specific to each individual one. So now we have information that's way more specific to the underlying fiber microstructure and the organization, independent of the other influences within the voxel. So from this, we can then summarize from the total information in the FOD, something like a measure of entropy, which captures the total information. If the FOD has a single well-formed peak, as we see at the top, entropy is low. But if there's a large number of peaks or very broad peaks suggesting fanning of fibers, then entropy tends to be higher. This work was followed by work by Till Rifford, also with MR tricks, where we can actually take the orientational information and measure the complexity in a very similar fashion. So we have two measures now that are natural alternatives to anisotropy and are valid in crossing fiber regions that give us essentially an inverse measure of FA, but that can be utilized for looking at the relative difference in the underlying crossing fiber populations at the voxel level. And when we look at FA in the mind sample and FA differences with age, here we see, as expected, the red showing older age is associated with lower FA. And then the bottom in the blue, a few regions where older age is associated with some increases in FA. But by and large, as we expect from the literature, we see this large negative age relationship. However, when we correlate this at the whole brain level with complexity, what we see is that here in the lightest blue represents the strongest negative correlation, that FA and complexity are highly negatively correlated throughout the brain, especially dominated in a lot of these crossing fiber regions. And if we take just these age-related, age-negative FA values and correlate those with complexity, we see that complexity is really explaining almost 2/3 of the variation in FA. And we follow this because the MIND study is not a representative sample, with work with the Berlin Aging Study 2, which is a population-based aging cohort study in Berlin, Germany, of younger and older adults, a subset of whom underwent MRI scanning. And this is work with students in my lab and Thad and John Plass, and with researchers from the Max Planck Institute in Berlin. And so in 102 younger and 318 older adults sampled from the population, and these are generally unimpaired. There is probably some incident MCI that's not fully well captured in this. We also have a cognitive testing battery. We're looking at just a few different measures that we composited into measures of speeded processing and fluid reasoning. And so we used the MR tricks constrained spherical deconvolution fixel-based analysis pipeline. I won't go into all the documentation. Most have followed their published approaches. But the key thing that I want to mention is that we measured, and this is a fairly novel thing, these measures of complexity and entropy for each individual case. And then we took the information from fiber density and decomposed that. So now we have images that represent fiber density just for the largest fiber population, and images that represent fiber density just for the second largest crossing fiber population. And we submitted these to a voxel-based general linear analysis of whole brain white matter, looking at continuous age and controlling for sex and headness and controlling for multiple comparisons. And what we found when we look at complexity as well as entropy is that older age is associated with both increases, shown in blue, and decreases, shown in red. So the regions in blue, there's higher complexity and entropy with older age, and red, it goes down with older age. And we see that even as a continuous measure that we're really capturing these differences in both systems when we sample the mean values from those regions. And even if we just look at the older adults, we still see a similar pattern, albeit more spatially constrained in the brain, where older age associated with increases and decreases in entropy and complexity. It's here we're looking at entropy, and we see that especially the age positive entropy region shown in blue, that we see is strongly correlated with older age. Now, looking at the fixel decomposed measures, right, the largest fiber population only, and when we look at age across the whole sample, we see that primarily older age is associated with lower fiber density in the largest fiber population, shown in pink, and a couple of regions shown in green where it's associated with increases. And that may be because of ventricular expansion, in part at least. But when we look at the second largest fiber population, we see regions in blue where older age is associated with increases in fiber density, and in yellow, decreases. So unlike the largest fiber population, there's more variable and heterogeneous change happening in smaller diameter fibers. And if we overlap those regions where we see decreases in the largest fiber population and increases in the second largest fiber population, well, suddenly that starts to approximate some of our changes in entropy that we see with age. And so regions where we see the largest decreasing and the second largest increasing is also where we see age-related increases in entropy. In contrast, where the second largest is decreasing and the largest is either decreasing or staying the same, that's where we see age-related declines in complexity and entropy. And when we sample from, and when we calculate a ratio of the second largest to the largest fiber population and sample from those same regions, it's explaining almost all of the variation, at least in the age-positive regions, especially in age-related entropy. We also looked at the relationships with cognition. And so higher age-related, age-positive entropy is associated with slower processing speed, the opposite for age-negative entropy, and the same pattern is manifest for a fluid reasoning composite. So Melinda Mirroring is a doctoral candidate in neuroscience, has been working in my lab for the past few years, and she's been looking at some of these data in relation as a function of white matter networks. So using graph theory structural connectomic analysis, where we can take whole brain white matter and in conjunction with free surfer gray matter parcellation and estimate a whole white matter network across the brain. And two of the measures that she's looked at include normalized path length or characteristic path length, a measure of network integration or the potential for information transmission, as well as Q-maximized modularity. And so modularity referring to clusters of nodes or modules that have more dense interconnectivity within them, but less or more sparse connections with other nodes. And she found that characteristic path length, again, the potential for information transmission, that higher path length is associated with lower age-positive entropy and vice versa for age-negative entropy. And the opposite pattern that we see for modularity. So older age is associated, tends to be associated with increases in modularity, but these are also associated with the age-positive entropy and age-negative entropy in opposite directions. The modularity was also associated with processing speed and fluid reasoning, negatively so. So older age seems to be associated with increases in modularity, but these are in turn associated with slower processing speed and poorer fluid reasoning, even when we control for age. So that's in population-based aging, but we were really curious, of course, how does this relate to differences in dementia, in mild cognitive impairment? How does it relate to differences in biomarker levels? And so this is where I've been really excited to work over the last year with some data from Nick Kanan's lab, his R21, to assay biomarkers from UMMAP participants. And so I think this slide becomes really redundant for most people here, but I'll mention Alzheimer's is neuropathologically characterized by a host of things, including beta amyloid plaques, hyperphosphorylated tau proteins, and neurofibrillary tangles, glial modulated inflammatory response, cortical hypometabolism, synaptic dysfunction. And we know that these processes can start 20 to 30 years before clinically diagnosable impairment, which is why I want to also show the JAK curves, which I think are probably, again, superfluous for this audience, but it's worth highlighting that, again, biomarkers from CSF, PET, and blood give us the ability to detect pathology well before the appearance of blue here of MRI findings in brain atrophy, and certainly then cognitive impairment that we see much later on. But I mention this in part because of work from Weston and colleagues, in which they suggest that if we can that measures of brain microstructure, shown in the yellow here, have the capacity to improve the ability to detect pathological changes with Alzheimer's earlier than more gross volumetric features. And so this perspective led to a few study questions that we reported this past summer at AEIC. So in non-demented older adults who have MCI or are cognitively unimpaired, are crossing fiber complexity and entropy sensitive to plasma biomarkers of Alzheimer's pathology? And are they comparable with tensor measures like FA? And are those things related to differences, the changes that we may observe in complexity and entropy related to cognitive performance? And so in a sample of 19 cognitively unimpaired and 29 diagnosed with MCI, including both amnestic and non-amnestic, but those two groups didn't differ on our relevant measures of age, sex, education, and race. We looked at plasma AD biomarkers from Simoa assays from the Canaan lab of beta amyloid 42, 40, and their ratio as a measure of relative severity of processes. And we did the same thing with p-tau 181, so tau phosphorylate at threonine 181, total tau, and their ratio. And that measure I had not really seen before, but I thought it worth exploring as a function of if that ratio should be capturing the relative severity of tau processes. And we controlled for handedness as well as MoCA score, and corrected for multiple comparisons and looked at this across all white matter voxels. So this is about 150,000 white matter voxels. So a lot of corrections for multiple comparisons and an increased tendency towards type 2 error. So there was a high likelihood we wouldn't see anything in a sample of 48. Nevertheless, we did find that higher p-tau 181 predicted increased white matter complexity in a region of the posterior cingulum bundle adjacent to the body isthmus and splenium of the corpus callosum. And when we sampled from those values and related the two, the p-tau 181 levels against complexity, we see that p-tau 181 is explaining almost half of the variation in complexity in the sample in crossing fibers. Even then, when we control for age, sex, education, race, and total MoCA score, we still see that strong effect. And we related complexity sampled from those voxels to a working memory composite of digit span task. And we see here again, even controlling for MoCA score, that higher sampled white matter complexity is predicting worse working memory even when we control for age, sex, and premorbid impairment. When we modeled entropy, we actually saw also effect in an adjacent but not identical region. So clearly, complexity and entropy are capturing somewhat different things that are happening in the tissue, and we see this in the fornix and in the isthmus and splenium of the corpus callosum most strongly, where higher p-tau 181 to total tau ratio is predicting greater entropy, again, even controlling for age, sex, and MoCA. And higher entropy in those voxels is also predictive of poorer verbal recall on trial 1 of the Hopkins Verbal Learning Test. Again, controlling for all of those same sociodemographic factors as well. So we know that toxic species of tau are known to affect oligodendrocytes and axons, and tau PET has shown in AD and even preclinical AD binding in some similar regions. We don't know if we're capturing end-stage tangles or simply something that's sensitive to early axonal damage and myelin damage that's happening as a function of increased toxic species of tau. But it's also corresponds with prior findings in voxel-based analysis looking at other ways of looking at biomarkers like total tau to beta amyloid 42 from work from Barbara Bendlin and colleagues. I also want to note that there were no significant effects or models from the beta amyloid measures or from total tau, and we saw no significant effects with our tensor measures, suggesting that actually modeling complexity and entropy it provides a far more sensitive measure of white matter changes that we see in early Alzheimer's pathology. Actually, I'm going to skip this for the sake of time. So we followed this up with some multivariate analysis more recently, and these are preliminary data from a work with Caleb Weissman, who's a second-year medical student at MSU in our Grand Rapids campus. And so looking at 109 cognitively unimpaired, 59 with amnestic MCI, and 39 with Alzheimer's dementia, we used partial least squares, a multivariate analytic technique, so that we can combine 4 different scalar images. So here we're using the decomposed fiber density, so separate images for the largest, second largest, and third largest. Instead of F1, we're calling them N1, it's the same thing, as well as a measure of extracellular cerebral spinal fluid so that we can get, or free water. So the idea being that if white matter is atrophying, there should be a corresponding increase in the extracellular compartment as well. And so by failing to capture that dynamic of change, then we may be missing out on some critical features here. And so by using these to compare cognitive unimpaired against Alzheimer's dementia and mild cognitive impairment, we hope to have a different characterization of these dynamics. And indeed, on the left here, we see our percent crossblot covariance, which tells us from these 4 different measures that we're using, how are they combining into different latent values. And so we actually see that there are 2 real latent values that emerge from these 4 different measures, both in their comparisons between cognitively unimpaired and Alzheimer's dementia and MCI. And when we look at the first latent variable, we can see what the loading of these different images is on it. And so we can see here in blue is the largest fiber population, in red the second largest, green's the third, and pink is CSF. And we can see that it's really being driven strongly by a difference in the largest and somewhat in the second largest fiber population, and that this pattern is also manifest in the comparison with MCI too, which is cool, because it actually seems to suggest some staging that we're able to observe. The second latent variable, however, is a bit more interesting. Here we're seeing regions where where fiber density in the second largest fiber population is lower in the unimpaired than in Alzheimer's patients, and that, or in those diagnosed with amnestic MCI. And so when we look at the brain images from these, for the first latent variable, we see these patterns here in yellow, where there's a higher, largest fiber population and somewhat second in the unimpaired versus Alzheimer's. When we look at the second largest fiber population, we see areas in yellow showing that it's the second largest fiber population has a greater signal in Alzheimer's patients, but areas in blue where the opposite pattern is observed, where the second largest fiber population, that fiber density is greater in the cognitively unimpaired. And one of the things we see is that some of these regions are canonical crossing fiber areas as well. What's more, when we look at that comparison with cognitively unimpaired and MCI, we see a similar pattern in the largest fiber population, but less so in the fornix for the second largest fiber population, but still maintenance of these changes in these crossing fiber regions that really mimic the pattern that we see in our comparison with patients with dementia. And Melinda has also been following this with network analysis, and here I'm showing you some work from our small world network analysis. And small world networks are those in which most regions are not neighbors of each other, but can be reached in a small number of steps. And it's also a reflection of network complexity. And it tends to produce networks that have properties of regional specialization and efficient information transfer. And we see that older age in our analyses is actually associated with increased small world networks. And this is very preliminary, by the way. We're still following this up with our collaborators because there's a lot of mixed findings out there and not all support this. But we see that older age is associated with increased small networks and that higher small network properties are also associated with entropy and complexity sampled from the regions where we saw the effects of tau and the ratio of p-tau 181 to total tau. It's not associated with education or MoCA score, but it is positively associated with hippocampal volume and marginally but significantly with the NIH Toolbox oral reading task, as well as the crystallized cognitive composite from the toolbox, even controlling for age. And with picture vocabulary score, but it's negative correlated with trial 1 verbal recall. So here's the question: is greater small world properties in non-demented people good or bad? And the answer would be yes, it seems, because we see both patterns manifest, but it calls to mind this notion that older age, we see these differential changes in cognitive abilities. So where older age tends to be associated with declines in fluid cognitive abilities like speeded processing and working memory and long-term memory, but increases across the lifespan in crystallized cognition like world knowledge and vocabulary. And so it really begs this larger question that we're trying to follow: what roles do white matter microstructure and network remodeling play in this? And when I see work like sending your grandparents to university increases cognitive reserve, this idea that ongoing engagement, not necessarily in doing crossword puzzles, but maybe in challenging and learning activities that really force one to not simply update some existing representation, but more from the perspective of accommodation and assimilation of new information. And this perspective from Aristotle that learning is an ornament in prosperity, a refuge in adversity, and a provision in old age, and a provision being a measure that we take beforehand to deal with some later emerging need. And so from this, my interest in the role of education and engagement. And so why does some of this matter? When we think about these claims about white matter integrity, our expectation is we have this single great thing that gives us a perfect representation of what's happening in white matter, when in reality, what we're actually seeing is something that is categorically different. But it's better in some ways because it's real, and because we can actually measure properties from the reality that we're able to now model and observe. And so rethinking white matter organization is this concept that white matter pathways are changing in response to both negative influences as well as to positive influences, and that microstructure and network properties are related to behavior, and that local changes in white matter are reflective of more global network changes that we can capture. But I also think it's worth, perhaps we need to thinking about redefining resilience, and perhaps it's the capacity for adaptive changes following either negative or positive effects. And all of this together is highlighting this idea of white matter dynamics as a potential marker and mechanism underlying cognitive scaffolding and reserve. And so has led to our emergent perspectives and questions. And this is where a lot of my work and funding proposals are now being directed. And questions like, does educational attainment create networks in the brain that support late life resilience? and then do subsequent formal learning and lifelong engagement systematically modify microstructure and networks. But can we disentangle these negative from positive influences becomes a really critical component of all this. And do these changes, or can they, in white matter microstructure and network, can they serve as markers of risk and resilience in cognitive decline in some translatable fashion? And I think part of this really necessitates thinking about additional components. So do adverse life course contextual or environmental factors moderate the effects of ongoing learning? And with that, I'm going to wrap up and thank my lab and students and my collaborators and everybody at the Michigan ADRC. I said to Stephanie at the beginning, I'm sharing these findings from UMMAP as if they're mine, but I don't really see them that way. I mean, the reality is I wouldn't have access to these data, much less the capacity to work with them if it weren't for everybody here and other people who've put in invested so much time and effort into UMMAP and the other studies and the ongoing efforts. Big thank you to Ben Hampstead, who's been really instrumental in helping me think about a lot of these questions, and to my rec mentoring team. Here's a picture of my students and I just before the pandemic. Thank you all for your time and attention today, and I'm happy to answer any questions.
Thank you, Drew, for a great talk. There's a lot of information to process.
It's dense. I understand.
Yeah, but there is to your point about the data between John and older adults and going forward in terms of things that might be affecting the white matter versus things that that may not. And you mentioned education. Something to think about maybe is the social context, not only measuring education attainment at years of education, because quality of education and the stress through lifespan, especially in children, might completely predispose the complexities or lack of in the future based on their development when they're children, a very younger age. So that, social account for social context in terms of a stressor,
I skipped over the slide. It was towards the end and I was running out of time, that actually really talked about those things. I mean, because, and especially Jen Manley and Maria Gleamore's framework for looking at kind of more intersections of risk and resilience and both life course factors at multiple upstream levels, as well as family and levels and these other contextual components, I think are absolutely critical. And so I totally agree. And that is a key part, I think, of what we need to be doing. But thank you. I appreciate that suggestion, Irving.
Hi, Drew.
Hi, Dave.
So I have two comments and a question.
Sure.
So my first comment is I personally think that the primary effect of age and Alzheimer's has nothing to do with pathology, but is solely due to resilience. And that with a decline in resilience with age, in a young person, in order to actually reach the threshold for cognitive impairment, you need much more pathology than when you're old. And that's because of resilience. So I think this is really very important stuff. A second comment I'll make is that the plasma total tau measures we now understand are contaminated by peripheral tau and are not very useful long-term in identifying what's going on in brain. CSF is different, but that there's an unusual form of tau that if you can measure that separately, you could discriminate between the brain-derived tau and the peripheral-derived tau in plasma. So I'm just adding that to your knowledge base.
I really appreciate it. I mean, that's exactly why I'm really grateful to have folks like you on the call, right?
But my question is the following. I mean, you're looking, it seems, solely at the microstructural content of the white matter. And, you pointed out how this precedes the macrostructural changes that are occurring in brain. But, obviously MRI is also capable of identifying total atrophy of a lot of these structures. And I'm curious to what extent, if you are combining both the structural and microstructural data into an index, and if you were to do that, would that perhaps show even stronger relationships, especially when you're including Alzheimer patients in the analysis?
I think that's a fantastic suggestion. I think Siri just thought so too. No, I have not been looking at that, but I immediately get it. And I'd be curious, to maybe chat a little bit more offline in terms of how to think about some of this in both a temporal sense, in that staging may affect that coupling between volumetric differences and microstructural differences. And I would need to think more about kind of which is more likely to precede the other. I would think microstructural changes would be should be more sensitive. And so then how best to include more gross volumetric changes to augment that sensitivity?
Well, I guess the way that I would think about it is, in some sense, what is the total white matter, burden, for lack of a better term? So you have the combination of having, fewer fibers within a certain volume of white matter, and that's going to increase. I don't know the details of what that's increasing, whether it's complexity or anisotropy or whatever. But the point is you've got that number, and then you've also got a number of the fact that that white matter structure is also getting smaller. And that the combination of those two is going to give you the total atrophy index. Structural atrophy is a very gross measure. But I think if you could combine it with this, I think there's an opportunity to really enhance and get a more complete picture of what is the true level of atrophy in that white matter complex.
I really like that suggestion, and I appreciate it. It's really already giving my mind a lot to think about and possibilities. And I say that in part because, atrophy effects, do you look in these kind of PAC regions, or are you focused on white matter atrophy versus cortical atrophy, or, regions that we know should be showing early changes? But I think there's a lot of interesting possibilities for how to think about and model those differences. But no, great suggestion. I appreciate it. And I'm happy to entertain further ones if they come to mind.
Okay, thanks.
Thank you.
Anything else before we close out today? All right, well, thank you so much for coming, Drew. That was a great talk. Thank you for being the first Leaders Initiative seminar in the seminar series.
I appreciate the invitation and the opportunity, and it's always good to be a guinea pig when the risk is not one that you'll be sacrificed.
Absolutely.
Take care, everybody.
Yep, bye.